Sir, YouTube start చేశారా? YouTube start చేశారా? Good morning, my dear participants. Am I audible?
Yes, madam.
Right. Good morning to all of you. We'll just wait for a minute. We are getting disconnected with a leap. We'll just wait for a minute. Duhita madam. Duhita madam, are you available? Duhita madam, are you available, madam? Am I audible? Duhita madam, are you available? Just please, all of you, be connected. As soon as we get connected from the Alip, we will continue. They're having a network issue, trying to get connected. Sir, Jaharullah sir, are you connected, sir? Zaharullah sir? Are you connected, sir? Please unmute, sir.
Hello, madam. Good morning. How are
Good
you?
morning, sir. Good morning, sir.
Yeah.
Sir, very good morning, sir, to all the participants. Good morning, Jaharullah, sir. I am very much thankful to you, sir, for accepting the invitation and to be a part of the virtual tour program, which is in coordinated with Alip Hub. I am very happy, sir, very thankful to you, sir. You have been very much supported um, for making
Yeah.
us or providing with the resource persons with domain expertise, which made our journey very fruitful in these 5-day sessions of our FDP, sir, and we will continue the same for the next week also.
Right.
Over to
Right.
you, sir.
Yeah. Yeah. Thank you. Thank you, ma'am. Thank you very much for that. And indeed, Gokaraju is doing a very good job, and this DST project will be helpful to all the faculty over here who have joined as the participants today. And in fact, for this whole 2 weeks program. And now we have today we brought this biotechnology tour for the participants to understand even in the life sciences, how the life sciences sector would be and what are the products that one can go for entrepreneurship and start up their company. So this particularly biotechnology unit of Elite Hub, which is an incubation center and the one which will provide access to the infrastructure and machinery for performing, the prototyping, product development, minimum viable product, and also the go-to-market strategy. So this biotechnology unit is a first of kind in India that has been, structured and uh, commissioned with the machinery uh, to get a complete end-to-end support for the startup companies who are into life sciences. So this particular tour is, as I told, to have an idea about the entrepreneurship life sciences, which is very much important because if you see here, we have many uh, business tycoons present in India who have their own companies in life sciences coming from diverse backgrounds, and they have been succeeded. So what you require is here, you just have to focus on a particular product and start on your own in the life sciences. So we have complete systems to support the life sciences sector entrepreneurs, and so that's how we are also ready. Elite Hub is always open for people to, to support and to make them their dreams come true in entrepreneurship and to become a startup as well. I hope Duhita is here. Duhita, are you there?
Yes, sir. Madam has joined.
Yes, Padma Garu,
Duhita
sorry
madam.
for delay. Good morning, everyone. Sorry, there's a bad weather, so we couldn't make it earlier. Sorry for that.
Yeah, Duhita, can you?
Thank you so much, madam, for you getting connected.
Thank you, ma'am. Thank you so much for the interest in everything. Just give me 1 minute. I'll show you around with the tour.
All right, madam.
Yeah, we will wait, Duhita. Right.
So our biotech students will explain regarding the equipments. I'll be there. Along with us, our students will explain them.
Okay, madam. Please unmute yourself, ma.
First of all, I
Joined
want
with
to
yes.
laminar airflow, where we isolate the microorganisms and please tell me.
Good morning, ma'am. My name is Sandeep.
Good morning.
This is laminar airflow, which attached with the HEPA filter means high free efficiency particular airflow. It can give the laminar flow means straight lines. The pure air will be come to the in this environment area. In this area, we can do the inoculation and petri plates and culturing. Done, ma'am. This is ultra low temperature freezer. It can use it for the preservation of wells and cells, some culturing cultures.
So actually, ultra low temperatures are used for the storage purpose only. So whenever we have a culture cells or bacterial cells that can be stored at a particular temperature, we can use this ultra low temperature. Few organisms will grow in a regular normal room temperature, right? To constrict the growing, we will use this temperature also at low temperature.
Good morning, ma'am. And this is called I'm Pranti, and this is BOD incubator. Now, incubators is generally for storing the cultures for a very long period of time. It has appropriate temperature conditions, moisture conditions, oxygen content, and everything is automatically set up here for the preservation and growth of our cultures, microorganisms. And then here we have shaker, orbital shaker. This shaker is generally used for uniform distribution of the nutrients and also for the gases exchange. And then here we have this apparatus, Soxlet extractor, which is generally used for the extraction purpose, like lemongrass extraction or any flower petals extraction for that extraction purpose. And then we have here fogger machines. Now these machines are generally used to create a clean sanitization and sterilization of the entire room. So this will spray the chemicals which are generally used for sterilization purpose, like formaldehyde and all those. And then we are very well known with this equipment. These are the microscopes. These are used to observe the organisms which can't be seen with our naked eye. This is used to observe the organisms which with their cellular level, Like we can observe the morphology, nucleus, and each and every cell organelle. And it generally magnifies the image. And then here we have spectrophotometer. This spectrophotometer is generally used for measuring how much a chemical compound, how much a sample absorbs the light, and used for measuring used for finding out the concentration of the samples. And then here we have pH meter, digital pH meter, which is generally used for measuring the appropriate pH, and it also records and displays the pH here. And it also got this electrode attached to it, where we put the sample here to find out the pH. And this is called as vortexer or vortex meter, which is used generally to mix the components of the sample, like to mix the liquid components to place here. And this is double distillation unit, and this is single distillation unit. These both are used to carry out the distillation process, through which we get pure water, which are used in laboratory for many purposes. And this is a weighing machine generally used to weigh the components. Here, the reagents which we use in our laboratory are usually measured in micrograms or milligrams. And this weighing machine works best for it, because through physical balance, through physical machines, we can't measure these minute quantities of the sample. And here we have water bath. So water bath is generally used for those compounds which are flammable under direct ignition flame. So here, to overcome that purpose, we generally put the samples in this, and it is operated at controlled temperature. And then we have hot air oven here. Hot air oven is generally also for the sterilization purpose. What it does here is, here we have dry air through which our sample sterilizes. And next here we have autoclave. As I said, hot air oven dry air is used here to sterilize the samples in hot air oven. Now we use autoclave, which uses the moist air. Like directly we sterilize the samples through the steam, pressure, and control temperature at 121 degrees, 15 bars pressures, and 15 minutes. We use this autoclave for sterilizing any equipment, culture media, and everything.
So good morning, everyone. My name is Vishal. So here is the hot room, which will help in the maintenance of all the steam generated for the useful of bioreactors here. So here is the bioreactor, we can see, which it is useful for the process of applying microbes growing the microbes in the large scale, like yeast or many microbial contents. We can make chemicals, biofuels, enzymes, proteins, and many pharmaceutical uses. So there, the black tank is a distilled water which will be used for this and making in a huge scale in large amount of production for the production purpose. So we've been working on this like lactobacillus projects, which I was working on lactobacillus, which is helpful to know the potency of lactobacillus which was available in the market, which I was working on the project, and my friends which were working on the biodegradation of the plastic, which is a very havoc situation right now in the plastic in the world, which is a very global warming. And my other friend which is working on the leachite, which underground water is polluted by the dump yards. So we are working, we 3 members were working on different causes and different things. So that's it.
Hello.
Uh,
Rita, are you
yes,
there?
sir. Hello, sir. Yes, I'm there, sir.
Yeah, can you switch on and then take it to the municipal solid waste tanks which are available there?
Which tank, sir?
municipal solid waste tanks, series of tanks.
Acha, acha. Okay, okay.
Yeah.
Just a moment, sir.
So these are the
Duita, I will explain you.
consumption
Okay.
of biofuels
Let
from
me-
the municipal solid waste that one of our startup is doing, who has received a grant from SIDBI of 23 lakhs.
Duita, I will take over from here. Okay. Hello.
Yes, sir.
Yes, sir, you are audible, sir.
Yeah, right. So can you switch on the video so that I will show me two things. One is reactor, bioreactor, and uh, yeah, bioreactor is enough, right? Fine. Thank you. Yeah. So this is an bioreactor, also called as a fermenter. It is about 500-liter capacity. So what to all the participants. So let me reiterate once again that what you are observing is an incubation center of biotechnology. So this incubation center is equipped with the machinery that comes with a very sophisticated and advanced machinery industrially. That's what I mean to mention. So not in the terms of R&D. So it's an MSME based incubation center for the first time set up in India, and it is the only incubation center that has this 500-liter capacity of fermenter or bioreactor. By utilizing this bioreactor or fermenter, you can produce n number of bioproducts. For example, you can produce probiotics. Presently, we have two companies who are in full-time incubation with us. One is Triton Biosolutions and other one is Harita Gold Enterprises Private Limited. Um, One particular company is producing, doing production of probiotics at large scale, and so they have been utilizing this fermenter, and they are supplying these probiotics to the aquaculture sector. And not only this product, but also you can also produce enzymes, antibiotics, proteins, vitamins, and biopesticides, biofertilizers, and bioplastics, raw material products, and so on. If you can say, you can even go for an crude extractions of any pharmaceutical drug products which are about to get into fermentation. So this machinery has complete upstream and downstream processing. You can perform upstream within nutrient media, and then we generally sterilize it by using the steam boiler. You can see on left hand side. Can you show that steam boiler, which will complete performs the sterilization of this reactor, which is an a double walled jacket, and you can see the complete the industrial scale machinery present over here. So you can utilize it for product development and MVP stage and go to market strategy. So that's what this whole setup is meant for. And so any company, any innovator who is interested to you know do perform some kind of prototyping or test your product with your idea or your innovation, or offered to perform some kind of trials over here, you can utilize this unit. So Atal Incubation Center, Elif, we have with biotechnology unit as the incubation facility is open for any innovator. Any non-science background people also can come, and it's open. As you know, entrepreneurship is open to all, so anybody can access or produce any kind of product and start a company. So considering that fact, so we want to invite you to come up with your own innovation in life sciences in any of the products. So we have set up this microbiology unit and biochemistry unit, molecular biology unit, and extraction unit, and then this is a fermentation unit. So in-house we have all these in one place of 1800 square feet, and these reactors and all the fermenters are available. So you have seen all the machinery over here also. So further we have companies who are we have students, we have research scholars, we have PhD people, we have industrialists who have utilized this product, who have utilized this machinery, and some of them have done their job works also, like fermentation of yeast, E. coli, and they have gone for an high production of antibiotics, and also some of the pharma drugs which works by using the microorganisms. So apart from that, we do have some innovators. Right now, we have a student innovator by name Shailaja. She is actually working on development of an bioplastic, and the plastic is biodegradable, and it can be utilized for any multiple customized applications in the industry. So she is very much successful in creating that bioplastic. That is one thing. So now I will give you one particular example of how to produce an bioplastic, which is referred as polyhydroxyalconate. So Duhida, can you take it to laminar airflow chamber? So show from outside, like both the laminar airflow and this ultra low temperature freezer both. Fine. So if you see here, suppose we want to produce PHA, that is polyhydroxyalconate, which is a bioplastic. What we do is we will get the cultures from MTCC, Chandigarh. Those are the bacterial cultures. The bacteria name is cyanobacteria. We will store that cyanobacteria in this minus 80 degrees freezer. On the right hand side, you can see it is a minus 80 cryopreservant freezer, industrial scale, and it was a very competent freezer that we have made and kept available in our incubation center. No any other incubation center will have this capacity. So we will preserve the cyanobacteria here, and then on the left hand side you can see industrial scale laminar airflow, which is an high specified specification machinery, not the lab scale what you see in R&Ds of academics and also in R&D labs, but this is an industrial scale. So what we do is here we will culture that, we will inoculate that bacteria, cyanobacteria, into the Petri plates, and from there we will take that Petri plates to the BOD incubator. Can you take to the BOD incubator? Next stage. So we have this BOD incubator where this cyanobacteria will be grown for a couple of days, 24 hours to 48 hours. The bacteria will be grown in the Petri plates. Do you have any Petri plates to show inside? Can you check? So these are the Petri plates in which the cyanobacteria would be grown, which produces the bioplastic. Fine. Now go to the flask culture. Flask shaker, rotary shaker. So we will inoculate again by using laminar airflow from this Petri plate into this flask. Each flask is about 500 ml to 1 liter capacity. You can put into these slots. About 80 slots are there. You can perform the culture. Almost it's a submerged culture and a broth culture and a suspension culture. It is called 3 types, and you can perform the culture conditions. It is run at certain RPM, about 80 to 120 RPM, and 150 also it can reaches. So You have an shaker rotary flask culture. We culture this like this openly as a vat, and we develop the bacterial cultures into some kind of product. A initial flask culture product would be obtained at this stage. From this stage, we will take this directly into the fermenter. Show me the fermenter now. Yeah. So cyanobacteria requires light also because it is a photosynthetic, and more modified rotary flask culture we will be using, not exactly what I have shown you. So for PHA, we use some light for the development of the culture of the bacteria, cyanobacteria. From there, the bacterial culture will be taken into this fermenter directly by using media, which is a nutrient media. And then we will pour the complete this inoculation of this flask into this into inside of this reactor. It is having 300 liters capacity of volumetric working capacity. And then after 24 to 48 hours, we will generally switch on this, and then we will monitor pH and temperature by using sensors. Show at the top side some more. Yeah. So here you can see some sensors which are pH and temperature sensors, and we measure the pressure also. So here you can see there is a motor on upper part. So mounted motor is present of 1 HP capacity. So this is to run. There is an right hand side blue color. So you can see that there is a shaft. So it is an impeller. Impeller will mix all the nutrients inside along with the bacteria, and it's a closed batch mode culture mechanism of a fermenter. After 48 hours only, we will take the cultures from the bottom. Show the bottom. So from the bottom, we will get the uh, complete product. You can see here, left hand side, there is an outlet. So there is another right hand side middle outlet. Actually, it is for to transfer the steam for sterilization. And the left hand side, you can see there is an outlet which is open, and through this we get the total culture with the bacteria. From there we will take it for the downstream processing. In downstream processing, generally we perform the extraction of the bioproducts. Now cyanobacteria produces PHA, which is polyhydroxyalconate. That polyhydroxyalconate will be taken into the filtration method. So generally we perform filtration, and then after filtration we will take out all the bacteria out along with solid particles of the nutrient media. We will get the liquid culture of the PHA, polyhydroxyalconate. From that we will purify it by performing crystallization. So crystallization is another method where we perform the purification of that product. So purification of the product would be performed by using crystallization. We have crystallizers. Uh, we have different crystallizers again, but we are not putting a complete machinery of downstream processing. Even we can able to get that machinery access from the ELIP. So ELIP is a total in nutshell. What I can say is, what you are observing here, this incubation center is located in ELIP Industrial Park, which is 32 acres of park with 180 companies inside. And this incubation center is available at one place within this ELIP main office, and we administer and manage the industrial park entirely. So it was set up by ELIP founders, MCM members, headed by President Ramadevi Madam. She is the person who started 30 years back this organization and also these industrial parks. Right now, 6 industrial parks are there in AP and Telangana. So, ELIP has a STP also, which is a effluent treatment plant. Very big plant is present in collaboration with the TSIC, T government, IALA department, and also ELIP combinedly. They administer and monitor manage the whole industrial park. So why I'm mentioning is because we have the 80 biopharma companies within ELIP, where we can get access of the machinery from here to any entrepreneur or the startup, and then we can get end-to-end support for the machinery, because all 80 biopharma units are having very high-end machineries. They are producing crude bulk drug production, enzymes, proteins, vitamins, and medical devices. You name the product that is available within this industrial park with all the industries over here. So ELIP have been supporting budding entrepreneurs, innovators, and startups like you, who can come and then utilize the facility at initial stages. So these are all the facilities and infrastructure. Apart from that, you can close, Duhita. Thank you very much. I will go with the information on funding. Okay. Right. So now, if you look into the funding, we have an MOU with BARAC, which is Biotechnology Ignition Research Assistant Council, Government of India, Department of Biotechnology, Ministry of Science and Technology. So BARAC MOU is present, and BARAC generally offers funding to all the startup companies in life sciences. If you talk and if you ask me what is the funding available for the life sciences, only one option I can give is BARAC grants, because it is an high level and very optimized and very much facilitated funding available for life sciences enthusiasts and innovators. Generally, we have CSR labs and many other labs in India, ICOR, ICMR, etc. All the professor scientists generally do research work and go with the publication. But right now, we don't want to end up your work into publication, but it should be commercialized. So what we mean is like it should be made into economical and utilized for our country. And if we perform startups to come into limelight, then there would be an increase in the number of jobs and economy of our nation, and also self development or individual development of the person. So three things will be covered at a time. So that's the reason we highly encourage you to become an entrepreneur. And now we don't want to limit your innovations into a publication, but it should be transformed into commercialization stage. So for that reason, now most of the grants available in India recently, last 7 years, they have been changed the policies, and they made it clear that most of their grants funding would be supported to the people who will perform the commercialization stage of the product, but not just an research that ends with the publication. So now we are giving funding to most of the people who does the commercialization stage of any innovation. So keeping in mind, BARAC has got 9 schemes. Out of that 9 schemes, one very efficient scheme is referred as BIG, Biotechnology Ignition Grant. It is about 50 lakh grant. This big grant is completely a grant amount, 50 lakhs given to you. You need not pay the amount back, but they perform due diligence and compliance is perfect that it should be utilized as per the submitted application and proposal. So that's what they see it for the next 18 months. You have to submit the proposal. For submitting a proposal, it doesn't require any fee to be paid. So you have to just have your innovation very clear. So you have to submit the application online. So the next call is opening in July 2nd, and any person, irrespective of your background in life sciences, I have seen most of the big grant applicants are non-life science background only. I repeat once again that most of the big grant applications are non-science background only. They are highly encouraging any person. Instead of you having, you need not say that I don't have a degree or knowledge, but you can have one person as a co-founder, or you can keep one person as a technical person into this field, and you can start a company as being the head of your own startup. So that's how we encourage and we take it forward. And there is no elimination and filtration for the people who come up with a non-life science background. We never filter them, but we highly encourage them to start their company, provided that they have a clear innovation and technical and scientific expertise within their team members. So this is what we consider here. Now, coming down the line, so Big Grant will give you complete missionary support, consumables support, and also a manpower and the working capital, and complete, you have an a complete working capital and also the capital infrastructure in terms of the fixed capital. You can get both at a time. Generally we referred it as recurrent and non-recurrent expenditure. Suppose say the recurrent expenditure would be manpower, where you pay monthly salaries. Non-recurrent expenditure is nothing but the machinery which you have purchased at a time, and you install that and utilize it for the next few years. So like this, for manpower salaries, for even the consumables in life sciences, we have consumables like raw material, and also the glassware and chemicals. These three comes as the consumables. Apart from that, we provide you rental also. You need to take a space of 1000 square feet. You purchase the machinery from this amount. About 30 lakhs out of 50 lakhs would be given for machinery purchase. You can have, starting from a simple, a small pin to an high level fermenter also you can install according to your project. So you need not spend even 1 rupee. You can have your own manpower, salaries paid for 18 months only, and then consumables as I told you, and whatnot. So I have covered you everything. So with these 50 lakhs, you will start with an small R&D to your prototype development. So prototype would be developed with your own facility with this 50 lakh grant, and then you would be further taken forward, where we have other grants available by name BIPP, SBIRI scheme, which goes up to 2 crores of grant rupees. Remember that grant means you need not pay back, but there will be a due diligence and compliance, provided that you should show the product seriously by floating a startup company with private limited, LLP, or a partnership firm. You should be a serious entrepreneur, then only they will give you the grant amount, because it is a public fund. Now, apart from that, what is the next stage? You will be given complete authority on the ownership of your own unit with infrastructure, machinery and all. And then from there, after 18 months of this big grant completion, where you got these 50 lakhs and you have completed your prototyping and you are ready with the commercial stage of your product, you will be given other schemes like BIPP, SBIRI, as I told you, up to 2 crores of amount. So there is no any ceiling amount for this ceiling limit for this BIPP, SBIRI. So it can go as much as maximum of 2 crores. It's a grant amount again. So you can establish a unit again of your own with some 10,000 liters of capacity, or you can say 50,000 liter capacity of your production. And you can have a rental space more, 10,000 square feet or 15,000 square feet capacity. And then you can expand your production as well by using these schemes. So likewise, I can say Bharat has 9 schemes which comes at different stages. At proof of concept stage, they have an big grant. At prototype stage, they have BAPP grant. At the product development stage, they have an SBRI scheme. They have MVP stage, they have CRN scheme, and also uh, they have PACE scheme at an MVP stage. Likewise, stage wise, they have a scheme which is very much useful to the innovator, provided that they have that clear scalability and viability of the product. And so these are the best opportunity we have an access for Barak team, wherein already our startup have got SBRI scheme of 25 lakhs back in 2018 itself. And Mr. Murli is the startup company. So we have about 15 startups in life sciences sector who are very serious startups. And we have different segments in life sciences. People who are in one, there are some entrepreneurs who come from computer science background, and they started into this clinical trials company. And we have some professors who are into the medicinal plant extraction products. Nearly 80 products they have. So even I mentioned you, some of the research scholars are there who are into biodegradable production, bioplastic, who have done their postdoc in different countries. 2 people who are there. They are into bioplastic production from PBA, which is an biodegradable compound again. So this is on the whole, I can say, I hope that it is audible, right? I'm just speaking.
Yes, sir, it
that
is
okay,
audible,
ma'am?
sir.
Right,
It's
right.
audible, sir.
So this is on the whole about this biotechnology unit incubation center of ELIP we have, which is located in Pragati Nagar of which is opposite to JNTU area. So any faculty or any person who would like to see and meet us at our office, you are open and you can come down to us anytime. You can just contact to our office, or you can contact Padma madam. She will refer us immediately, and then we can discuss on whatever the idea you have in life sciences sector. So we have many other grants: METI grant, SSFS Startup India Seed Funding Grant, and AAM Atal Innovation Mission Niti Aayog Seed Fund Grant. And so we have these seed funds and the grants available, which we can able to identify potential startup, and then we can fund them and support them in their venture development. So this is on the whole in nutshell, I can say. So if I have any questions, I'm open to take it from here. Yeah, thank you very much.
Thank you, sir. Thank you for your deep insight into how the actually this biotechnology lab has been established, and stage by stage how it is been used for the production. It was a really wonderful session. Hope all the participants might have got the practical exposure in this virtual tour, how the industry is and how we should be able to see that our students are also get involved into this entrepreneurial aspects for Make in India. It very clear explanation has been given, sir. Sir, one question from my side: is there any time slots will be allocated if, say, for suppose for biotechnology startup, if there are 5 groups are been coming in, and you will be allotting some time slots for them to work with the present technology which is available in this Ivar Alip, sir?
Right, ma'am. So we will give slot-wise access to this facility. Those who want to do prototyping and also product development, and then they can have a complete access. That is one way. And second way is we will generally try to apply for an big grant fund. Through that, they can get entire setup on their own also. So that will be done simultaneously with the access to this lab. So lab, I won't say it's an unit, which is an incubation center come industrial scale MSME based setup. So it's not an R&D totally which will work like you can find scientists over there. It is not an for academic purpose where skilling and training would be done. So it's an innovation unit and more industrial scale production testing into the market. And also, you can do prototyping, you can do trial and errors, and you can actually, you can validate your product as well. So for validation of your product, so we give an slots, and then you can take up the access at very nominal rates. And even we can able to sit down and we can able to mentor you further how it can be accessed, and also how we can able to support you as well. Yeah.
Thank you, sir. Is there any age limit for this
No age limit,
aspects?
madam. No,
Yeah.
not at all. Yeah, that's a
Okay.
perfect question. From 18 years of age up to 70 years of age or more than that, any person
Okay.
who is interested, irrespective of age, gender, I'm telling you gender also, because you may think that it is only for women startup. No, we are open to all. Okay. So in fact, there are certain provisions for the women being the ELIP VHub and ELIP organization that will be given a priority. But we say that, if a startup is a women startup directly, that is well and good. If it is not an women startup, we call it as women-led startup. Men can be, men can be the leading or face or the front face, and but provided that there should be shareholding to the women within their company. It can go up to a minimum percentage up to a maximum of 51% of shareholding. So it is called women-led startups or enterprises. So for that, we are open, and so men also can easily have the access. We are open for men as well. So irrespective of gender, age, and qualification also we won't consider. We have people who are just seventh class, and we have people who have done their postdocs, double postdocs also, and PhDs, postdocs in different countries, came back, and then they started their own life sciences products. We have given excellent support to these people, and including so entrepreneurship is open. So considering that principle, so we have case studies, we have perfect success stories also in ELIP. Yeah.
Sir, apart from the BIRAC, if we get funding from DST or MSME also from the incubations and all, we can able to work here, sir, using your ALIP
Yes,
infrastructure.
exactly. Any innovator who can have an product and who have an innovation idea also, at least idea also is enough. We can fund them even in other grants also, like MIT, as I told you already, MIT, Startup India Seed Fund Scheme, AM Seed Fund. We fund them in this through these schemes also, and to any life sciences startup. See, we have one startup into medical side, into biomedical equipment. We have Jeremy Ban, who is a successful startup. They have an sterilizer that kills the microbes in air and surfaces, that is used in surgical theaters, operation theaters of hospitals. Apart from that, in all diagnostic labs, and also anywhere in offices also. So it sterilizes the whole room. It's a very a high level, very successful product, I can say. It got 40 lakhs grant from us, even 7 lakhs grant separately. So it has got a market connect, huge market connect, and it has got an net worth of right now 22 crores. The company has reached. It is called as New Inventronics Private Limited. So we have done experiments in our biotechnology lab on bacterial cultures during pandemic stage. So even our biotechnology lab conducted our personal, along with me and Duhita, we have done experiment in COVID isolation ward also. You believe me or not, we have done. We when you people all sitting in home, the whole country, when the pandemic was there, uh, I'm talking about 2021
21, 22.
exactly March 31st.
Yes, sir.
Okay, we got around COVID lockdown 29, I guess, right?
Yes, sir.
So 31st March, we took permission from Government of India, and we did experiment in isolation ward of chest hospital by using this sterilizer. And myself and Duhita is also there along with me. And so we did this test, and then we found that it is doing 100% results, and we've tested that in CCMB BSL3 lab also on coronavirus later. Then we dedicated to the nation. It was inaugurated by the Home Minister of India. So this product got wide acceptance in India as well. And so that's how this is an startup from our female startup, women startup by name Shirisha. She's the innovator of this product. Um, so she has 20 years experience in biomedical equipment, design and development of different products. Now she came in with that product within a month by doing fabrication day and night, and so that's the best product from this lab we have supported to the nation. Apart from that, I can take many examples, and we have a lot of things. In fact, like I will take a small session on Monday. I will introduce you our few of our 4 startups are coming, who are very uh, you know serious and very innovative products, serious startups with an innovative products, and ongoing the companies with who are expanding themselves, who have received the grants, who are highly into the market penetration, go to market, and So with attraction also in their companies, you will see 4 best startups on Monday. And I hope like you should not miss this session. So they will be introduced by me at morning 9:30 a.m. Right?
Yes, sir.
So we will see these 4 startups, madam, with the different products and also portfolios and their products. I mean, the startup companies and their business and market stage and everything you will see from their own from their own the founders itself. I can say the founders they themselves will be speaking on that day. So this is what the session is. I hope like if I can get any question from the participants, I'm happy to answer as well. Yeah.
Yes, sir. Requesting the participants, if you want to pose any question, you can post in the chat window, or you can just unmute yourself and have a question. Any question related to the startup and Alip we have? Participants, please respond.
I think they don't have any question. It's okay, ma'am. So even they can reach out to us.
Yes, sir.
Duhita, are you there? Can you share our email ID, our we have email ID on the
Yes,
chat
sir.
box
Yes, sir.
so that they can
Yes, sir.
continue. So thank you very much to one and all. So with this, I hope you have got a good insights into this through this virtual tour and the information to you people. So we thank you for giving this opportunity. Right.
Thank
Ma'am,
you,
shall
sir.
I exit?
Yeah.
Yeah. Thank you very
Thank you,
much.
sir. Thank you so much, sir.
Yeah. I think we have next visit by T Works, right?
T works in the afternoon, sir, by 1 o'clock.
By 1 o'clock. Fine.
Yes,
So
sir.
we will. I will also join. So I will
Yes,
be
sir,
as a
definitely.
participant. Just I will observe. Okay. Right.
Yes, sir, definitely, sir.
Right.
There
Thank
is
you
one
very much.
question.
Right. Thank you.
What are the safety measures taken? There is one question.
Asha, there is a question. Okay.
Yes, sir.
So what are the safety measures taken? Fine. So so the question is very generic, and I can
Or
give
these
you.
healthcare products, sir.
Sorry, which product?
Healthcare products.
Healthcare products. See, one is as I told you, this Jeremy ban, it comes with an UVC light, which is very harmful, and it causes skin irritation, and also down the line, if you get major exposure of UVC at 256 nanometers, it will create skin cancer also. So we close the complete chamber or the room of 1000 square feet, and then we sterilize it for only 15 minutes, not more than that. Even you can do it from 5 minutes to 15 minutes of that range, and then you have to open the door and then you have to come inside. It can be switch on and off by using a remote outside the room. So it's a remote controlled device. So in that way, you can switch off and come into the room. Now further, the company has made a hello, hello, hello. Am I
Sir, you
audible?
are unmuted, sir. Sir,
Hello.
yes.
Hello.
Yes, sir.
Am I audible, ma'am?
Yes, sir.
So I think like I just I started explanation. I think you people have not heard, right? Because of
Yes, sir.
my mute, right? Fine.
Voice.
Let me speak once again. So we have regarding the safety measure, for example, to this Jeremy ban. The Jeremy ban unit comes with an UVC with 250 nanometers of UVC intensity, and it is very dangerous in terms of skin. It causes skin irritation, and also uh, if the exposure increases, it goes to up to skin cancer. So what we do is we close the room. The room should be 1,000 square feet, and we use a remote, and then we switch on for 15 minutes, and then we switch off after that. So from 5 minutes to 15 minutes exposure or switch on of this UVC along with other integrated components, it was patent granted for this device. And it has such a mechanism, like, even if you unknowingly anybody enters, open the door of that room and enters inside, immediately it will cut off and switch off automatically. Such a sensors we have kept. So that's the safety we have provided, which is the best safety method. And further, as I told you, to remote, you can switch on and off. After 15 minutes, you can get inside by switching off the machine or the instrument. So this is the safety that we have provided it with. And now we have got IoT based product also, where updated and upgraded. You can monitor by using mobile phone. You can increase, decrease intensity, switch on and off. And of course, we have kept open for this regulation, wherein if anybody enters inside, automatically it switch off with the sensor. So any mobility it senses, and then it can switch off. So this is what the safety we have done for this product. Yeah, I hope I have answered my question, right?
Yes, sir. Yes, sir. Thank you so much, sir. Sir, is there any medical council also get connected with this, any of the medical products to get certified and used for the human applications, sir?
We have many regulatory agencies and
Yes.
licenses for which, one has to get the product approval. One is we have DCGI,
Okay.
Drug Control General of India. That is one, which is having one body by name CDSCO. So CDSCO gives approval for any machinery. One has to give this approval. But when we contacted CDSCO, they mentioned that it is not required for this machinery because it is a sterilizer, and still we can use that machine. So we got no objection from them, from CDSCO. So CDSCO is an DCGI body, Government of India. Apart from that, one can have an licenses and standard and quality approvals also. ISO is the best one where we can say. And next one is one can have any electronic based approvals also. So CE is the one which we get the approval. You can have a CE approval also. And there is one medical regulatory body which is present in Chennai, which gives medical devices approvals, and they generally validate the product and they give the certificate also. So you can have an certification from that body, and it comes under again the Government of India, Ministry of Health, and also IMA, Indian Medical Association as well. If you want to place any product into IMA, into hospitals, then you should get the approval from IMA. There is another body by name NABL, National Accreditation Board of Laboratories. Even NABL accreditation is not necessary for this because you are not getting any lab. But in fact, if you have your own startup with a lab, R&D lab, which requires an CRO to be done, like contract research organization, if you want to perform for your lab, then it requires NABL accreditation for such labs. So that's quite different again. I just mentioned because, as you mentioned, the regulatory bodies and licenses. Yeah.
Thank you, sir. Thank you for your wonderful session. Thank you so much, and Duhita madam also, along with our journey of this FDP for a 2-week program to get associated, to get a lot of information as a practical exposure, sir. Thank you so much, sir.
Thank you, madam. Thanks.
Thank you, sir.
That's great. Yeah. Let me exit, right. Thank you very much.
Thank you. Thank you, my dear participants. You get connected by 12:50 in the afternoon section to get more information about the T-Works, how the co-work spaces are available and how we can utilize for any of our entrepreneur activities. Thank you all.
Thank you, ma'am. Thank you.
Thank you, madam. It was really a wonderful journey towards the Alif We Hub, madam.
Thank you so much for the opportunity, Padma Garu and Gokaraju Rangaraju Engineering Institute. Thank you all.
Thank you, ma'am.